Hello all. Myself, Dr. Pratima from Department of Microbiology, and our today's lecture is on MB 361 Medical Microbiology. In that, we are going to study our second credit, that is animal viruses. In the animal viruses, two viruses are included. First one is foot and mouth disease virus. So we'll start with this foot and mouth disease virus. About this foot and mouth disease virus. It is highly contagious. This is a disease of cloven hoofed animals. You can see in this figure this cloven hoofed pattern of animal, and it is probably more infectious than any other disease affecting animals and spreads rapidly if it is uncontrolled. It affects cattle, mainly buffaloes as well as cows, as well as sheep, pigs, and goats. Mainly, these domestic animals, they are highly susceptible to the infection by this viruses, foot and mouth viruses. But other animals which are not a domesticated one, in that you can include elephants, they are also highly susceptible, and other any type of a cloven hoofed animal. Rats are also susceptible to the infection by this. About this disease, here initially we have to study the diseases of human beings like respiratory viruses, hemorrhagic viruses like dengue, hepatitis A viruses, gastrointestinal viruses like rotaviruses, cutaneous viruses, some neurological viruses like encephalitis, Japanese encephalitis viruses. But these are mainly diseases of human beings, while these are diseases of animals, but sometimes these diseases may be transferred from animals to the human beings, like now you can call it as a zoonotic type of a disease. So here what we are going to study in this foot and mouth diseases, that is the virion, that is causative agent of the disease, their characteristics, their viability characteristics, then the pathogenicity, that is the property of causing disease, and the pathogenesis. Pathogenesis is nothing but the manner of development of disease. Then the symptoms, clinical symptoms when infection acquired by the animal. After that, we are going to study laboratory diagnosis. In this, not only isolation of viruses included, but serological diagnosis also. Then epidemiology, that is how this disease spreads in countries. And lastly, prophylaxis and chemotherapy. Prophylaxis, that is how to prevent these infections, and in the chemotherapy, treatment is included. Now, in detail about virion characteristics. So here this foot and mouth disease virus is belonging to the aptovirus. Aptoviruses, this is nothing but a genus belonging to the family Picornaviridae. Pico, that is small, RNA, that is RNA viruses. So name given to this family, the Picornaviridae. The FMD viral genome consists of a single strand of a positive sense of RNA, and it is approximately 8,000 nucleotide bases having RNA. And this virus, as you know, is very small, that is 25 to 30 nm, and so called as a picovirus. It is spherical, having icosahedral symmetry, and one capsid encloses whatever the nucleic material of this virus. In this figure, you can see this capsid is consist of 60 copies of capsomers, and each capsomer is composed of 4 structural polypeptides. So that polypeptides are labeled as VP1, VP, and VP4. Now, regarding various other viability characteristics of FMD viruses. This is highly temperature sensitive virus and rapidly get inactivated at elevated temperature at a high temperature, as well as this one is sensitive to pH also, mainly elevated pH. So viral survival is basically between pH 7.2 to 7.6, that is at a neutral pH. But in acidic as well as in alkaline condition, it easily get destroyed. So here inactivation occur below pH 6.5 and above 11, that is in alkaline condition. So this virus can survive in dark, in moist or humid conditions, but rapidly inactivated when there is a desiccation occur, that is low level of a moisture in the atmosphere, as well as you know pH and temperature sensitive virus it is. It can survive in milk and milk products. So upon ingestion of such a type of a contaminated feed, animal may get infected. Then, regarding serotypes of these viruses. So, antigenically, it is heterogeneous. Heterogeneous, various types of serotypes. Total 7 serotypes are considered important. So, they are recognized as a serotype O, serotype A, serotype C, SAT1, SAT2, SAT3, and Asia 1. This SAT is for Southern African territories, as the initial outbreaks mainly occur in the Africa, in South Africa, because of these variants. Each serotype of this foot and mouth disease is antigenically distinct from each other. This is very important characteristic we have to consider in the preparation of a vaccine for the disease FMD, because whatever the antibodies developed against these serotypes, they are not cross reactive. So every time you have to use separate vaccine for that particular strain, because of which outbreak occur. So here each serotype is considerable, having considerable antigenic variation, antigenic diversity. For this reason, antisera raised against one strain of a serotype may not recognize other strains of a same serotype. So this is important for preparation of a vaccine against these viruses. Now, we'll start with how disease occur in the animals. So basically, mode of transmission, animal transmission, occur by two ways. There is either direct transmission or a indirect transmission. In the direct transmission, it is mainly by inhalation. Whatever the aerosols containing these viruses, which enter in the air from infected animal only, they travel long distances, and when proper temperature and humidity present at that time, they may travel long distances and infect other animals in the herds. This is one way. Second way in the direct contact is because of a direct contact with the vesicular fluid from the infected or a ill animal or a sufferer animal. Here, ingestion of infected animal products, infected contaminated milk, and milk products also responsible for the occurrence of infection in other animals, including human beings. Next one is the indirect transmission. In the indirect transmission, contact with the contaminated food is one reason. Then here the cattle trucks, lorries, they also transmit this infection from one animal to other. Then marketplaces, loading ramps, as well as the roads. Uh, Here mainly the uh, whatever the vesicular fluid which contaminates soil is one cause of a spread of infection. Then the fomites, the inanimate objects like people, other animals, they also transmit this infection. And the mechanical, that is the milking person, may transmit with the help of their hands these viruses from one animal to other animal. So these are various ways of transmission of viruses from one animal to other. Upon entry of these viruses, either by inhalation or by ingestion or because of a direct contact with the vesicular fluid, these viruses now enter in the second animal. Here, upon their entry, if it is by inhalation, the respiratory tract, mainly upper respiratory tract, in their epithelium, these viruses penetrate, they invade, they multiply, and after that, they enter in the lymph nodes. In case of ingestion, the viruses enter in the gastrointestinal tract. Again, they enter in whatever the lining of a gastrointestinal tract, and after that they enter in the nearest lymph nodes, and after that their entry into the blood circulation. So when viruses entered in the blood circulation, at that time there is a high grade fever, that which is the first symptom of a disease appear. So here after that, as you know, blood circulation is present in each and every part of the body. These viruses reaches to the peripheral blood circulation, and after that there is a vesicle formation. You can see on the skin, vesicle formation. So this type of a vesicles are mainly observed the foot region as well as in the mouth parts of an animal. So these vesicles are formed within 1 to 4 days when severe infection by highly virulent organism, highly virulent viruses. So vesicles are mainly present on the lips, that is on the oral mucosa as well as on the foot pad. So this one is the second symptom of the infection, pyrexia, that is high grade fever as well as a vesicle formation. So this is the description of how there is a formation of a vesicle on the mainly of a lips as well as on the foot part. So this viruses when enter in these parts of a body, at that time that vesicles get when vesicles are formed, vesicles get filled with the fluid, and because of which difficult for that animal to feed now. So this one is a part of a primary viral replication after inhalation as well as after ingestion. This takes place in mucosal as well as in lymphatic tissues as well as lymphatic tissues of the pharynx. Viremia, that is high grade fever, follows primary multiplication, and further viral replication in the lymph nodes, mammary glands, and other organs in that mouth, muzzle, teeth, interdigital skin, and coronary band is included. In these areas, why infection mainly spreads in this area? This is because of a stratified squamous epithelium, the cells of which are mainly host for these viruses, and vesicle formation results in swelling that area as well as a rupture of a keratinocytes in the stratum spinosum. So this is part of a pathology. We have to study only pathogenesis in this. And now, after outbreak, after multiplication of the viruses in different parts, you can see various types of a clinical signs, that is symptoms. So after 2 to 14 days, depending upon virulence of that organism, of the viruses, as well as immune status of the animal, can see incubation period. Now, here in the initial stages, there is a shivering, which is the primary symptom, and very high grade fever. High grade fever, that is up to 41 degree Celsius, or if you check it in Fahrenheit, at that time 104 to 106 degree Fahrenheit temperature observed in the affected animal. There is a anorexia in the animal. Anorexia means loss of appetite. They unable to feed during unable to ingest food during this condition because of a large vesicles in their mouth parts. There is a continuous slobbering in the next day. Slobbering means nothing but there is a plenty or profuse salivation, where saliva hanging in long ropey strings up to the ground, and because of which, okay, when the movement of their lips occur, at that time a smacking noise, smacking lips, it can be observed in the animal. Because of this infection, some other effects are also observed, in that reduced milk yield is one more symptom. Then, when vesicles are present on feet, mouth, nares, muscles, teats, the condition of that animal become worse. That animal unable to stand on the feet because of presence of such a large vesicles. This vesicles progress to erosion, and this leads to lameness. That animal unable to stand and also become reluctant to move because of a painful condition. There is a slogging of hooves occur. You can see in the next figure how that slogging of hooves is there. In the pregnant animal, abortions are very common during the infection, and in young animals, in cows, A mortality rate is observed very high. So death in young animals also observed as a complication. So here in this figure, you can see symptoms of the disease. That is in the lips. On the lips, there is a formation of such a type of ulcers. Okay, upon breaking of vesicles which are formed in the secondary stage of infection, as well as on the foot pads, as it is infection of a cloven hoofed animal. Here in this figure, you can see how there is a necrotic type of a lesions are observed in that. Then in the next slide, you can see other symptoms also, which are mainly observed in sheep. Sheep are also cloven hoofed animal. So there is a sudden severe lameness in sheep. There is a tendency to lie down as they unable to stand. They reluctant to move when made to stand, and blisters are mainly present on the hoof and mouth. Again, there is a anorexia. They unable to eat, as well as loss of appetite is one more symptom in case of a sheeps. In case of a pigs, similar type of a symptom, but they are more severe than cattle's. Highly painful condition. There is a formation of a coronary bands in the interdigital spaces also. The animal become lame. There is a snout vesicles, oral vesicles, which are less common in pigs than the cattle's. Then, in the next stage, we'll see after pathogenesis, that is, after appearance of a clinical symptom in the laboratory, how to identify that the animal is suffering from foot and mouth disease. So, first step in this case is collection of a samples. After collection of a samples, here requirement is to identify what type of symptoms are there, and based on the symptom you can collect samples. Then there is a next step in the laboratory diagnosis is initial diagnosis, which is symptomatic only. Then isolation of viruses from the collected samples, then identification of a samples in the laboratories using molecular methods like ELISA, RT-PCR, complement fixation test, then viral neutralization test, like this test are used. This is part of a virology. So here the first step in the laboratory diagnosis, what to collect. So here you can collect various samples which are readily available in the infected person, in the infected animal. So in that epithelium and a vesicular fluid which is readily available, as it is present on the lips as well as on the feet of an animal. Then one can collect a blood sample from which you can separate serum also. Then oropharyngeal fluid, which is called as a probank, can be collected as a sample for the laboratory diagnosis. But in case of a fetal animal, upon death of the animal, yeah, in the epidemiological survey, one can collect some parts, some organs of the animal. So, which are they? That heart of young animal is collected. Then, from the lymph node, in that prescapsular in the ruminant, submandibular from pigs, and mesenteric case of all types of species, one can collect material. Then spleen, kidneys, as well as a liver, which is after autopsy only. You can collect material and check it for presence of a viruses. That. So here that star is for fetal cases. But in case of a living animal, one can collect mainly vesicular fluid and whatever the material, exudative material present on the fits of the animal. So, after collection of these samples, a number of tests can be carried out. So, which are they? So, in the laboratory diagnosis, mainly two main methods are there. First one is virus detection method, and second one is for serological surveillance. This one is mainly carried out in the epidemiological survey during the disease outbreak. So we'll see first part of a laboratory diagnosis, that is a virus detection method. So in that, first one is antigen capture sandwich ELISA is the first method used for for identification of these viruses. Second method is PCR based method. In the PCR based method, multiplex PCR, which is MPCR, can be used, or RT-qPCR can be used. RT-qPCR is nothing but a quantitative reverse transcription PCR. So this can be carried out. And third method in the laboratory diagnosis is isolation of viruses and subtyping of the viruses. Using all these methods, you can identify these viruses because only seven serotypes are there. In the serological surveillance, which is part of epidemiology, virus neutralization test is carried out, or the complement fixation test is carried out. Liquid phase blocking ELISA LP is one more test included in this serological surveillance. Then solid phase competition ELISA SP can be carried out in this. So, this serological surveillance in the epidemiological survey is carried out for two main groups of animals. First one is vaccinated, and second one is the infected. In the vaccinated one, structural proteins, that is SP, they are identified. While in the infected one, non structural proteins, that is NSP, they are mainly identified. Now, the main part of the lab diagnosis is of a isolation of viruses. So here, BHK21 cell cultures, they are mainly used for isolation, propagation of these viruses. So these are nothing but a cell layer of baby hamster kidney, BHK21. In these cell lines, you can inoculate material from that vesicular fluid from infected animal, zero point 0.2 ml of that clarified suspension of epithelial tissue samples you have to inoculate in them. After that, one can see within 48 to 72 hours, there is a cytopathic effects in the cell lines. So CPE can be examined after 48 hours. Now, here there is a formation of a some type of pockets, plaques, okay, where there is a clearance of the epithelial lining upon inoculation of the viruses within the cells. After collection of whatever the material from these affected cell lines, one can perform two more tests. First one is a virus neutralization test, and second one is a complement fixation test. In the complement fixation test, sedimentation of a hemolytic system indicates positive result. That is, the same viruses are present which are responsible for foot and mouth disease, and negative results, that is, there is a full hemolysis of the serum samples, indicate negative. So one can record results accordingly. Then next part is of a prophylaxis, that is, after infection, after laboratory diagnosis, how to prevent these infections in the animal. So this one is very important during the epidemics, that is, the infection must be avoided, as it is very easy for these viruses to transmit from one area to other. Here, stamping out is the main step. Stamping out means destruction of an infected animal, or the you have to sacrifice that animal. One can use euthanasia. Euthanasia, which is again method of killing that animal, but it is painless killing, where the suffering animal, which is not curable one, uh, to avoid that pains in that animal, one can use this method of a euthanasia. that animal afterward enter in irreversible coma. So it is of no use to treat that animal. In that case, okay, this method is used to prevent infection in other animals. Then tracing play very important role in this case. That is immediately identify infected animal, quarantine of that animal, that is separation from other animals, and you can use number of a combined type of a strategies to prevent spread of infection. So here, in that proper treatment, proper disinfection of the premises is included, as well as proper destruction of a that animal using various methods, in that digging etc also included, digging off that particular cadaver, whatever the litter, whatever the excreta of the animal, as well as animal products. Incineration is also one more method in the combined type of strategies. Then movement control is also included in this category. Movement control, that is, the animal should not be transported to some other places. Infected animal should not be transported. So strict import restrictions, this is mainly by legislation only, they are required. Then one should prohibit live ruminants, swine, as well as their products from FMD affected countries to other countries. Here, one more type of a prophylaxis is included. That is, in case of a mainly for pigs, heat treatment of a swill or whatever the garbage which is fed to pigs. Because of that, also transmission may occur. And monitor that disease outbreak worldwide, because it is very easy for these viruses to move from one area to other. The main part in the prophylaxis is of a vaccination. So here killed vaccines are available, which are highly serotype specific. As you know, there are total 7 serotypes. Accordingly, based on that, one have to prepare vaccine and vaccinate their herds. It is essential to isolate a virus during epidemics and identify the serotype to select correct vaccine. There are a number of vaccination issues. There is a requirement of a annual revaccination, which is time consuming as well as a costly method. But one more problem in vaccination, it does not protect against infection from other serotypes of the viruses, but it reduces clinical signs. And during that condition, viruses is spread from one animal to other animal. So here, based on this, in the epidemiology, the distribution of the disease one have to study. Virus transmission, how it occur and how to control it. So as you know, aerosols as well as a contaminated feed is the main source of the infection. Then here susceptibility of the animal to the infection, that should be controlled. The carrier state is observed in the animal. So here there is a tracing, there is a screening of the animals in the herd is essential. As you know, there is 100% morbidity after infection in the susceptible animal. One have to take care of the animal population. Initially, this disease observed in South Africa, but afterward it spreads to number of Asian countries also. But in US, in Canada, and in Mexico, in pigs, higher outbreaks of FMD observed. Mortality rate nowadays is reduced to less than 1% because of a continuous vaccination in the herds. There is more chances of infection in young animals. So during that stage only, vaccination is essential. Animals which are affected should be destroyed to prevent spread is again part of epidemiology. Now, last part is of a treatment to the infection. So there is no specific treatment after infection to the animal. So there is no immediate treatment once the animal is infected. So infected animals should be isolated, that is quarantine is again considered as a part of a prophylaxis as well as of a treatment to other animals. And here symptomatic treatment may be provided to them. There are formation of a number of a mouth lesions in the early stages of a disease. So one can use 1% potassium permanganate solution as well as a boroglycerin, which is boric acid glycerin paste, can be applied to the vesicles which are formed or the mouth lesions which are formed in the affected animal. In case of a foot lesions, these foot lesions can be washed again with 1% potassium permanganate, or one can use 0.5 to 1% baking soda, which is nothing but a sodium bicarbonate, and one can apply antiseptic cream on the lesions, on the ulcers which are formed. Then clean the animal shed, avoid infection to other animals. Proper disinfection by using disinfectant is one more part of a not only prophylaxis but as part of a treatment. Now there is a problem in number of countries where the disease is endemic. At that time, secondary infections by bacterial pathogen is one more reason for the fatality in the animal. So in such a endemic countries, antibiotic therapy, that tetracycline is mainly used as a treatment. Antibiotic therapy used to control secondary bacterial infections of a ulcers, because these ulcers are nothing but open wounds, and as you know, in water as well as in feed of animal, number of bacterial pathogens are present which may contaminate these lesions, these ulcers, and the conditions are become complicated one, worse one, initially viral infection and secondarily bacterial infections. So, tetracycline like drugs are used in the treatment of this secondary infection, but it requires several months uh, for the proper recovery in the animal. So, we can summarize this foot and mouth disease. Okay? That is, it is highly contagious viral vesicular disease of cloven hoofed animal. It is mainly the disease of a livestock, important livestocks. In that, pigs are included, cattle, sheep, as well as goats. Mainly, it affect the economic conditions of a number of countries during such a outbreaks, during such a epidemic. There is a reduction in the milk yield as well as in the meat yield. There is a problem to the hide industry because of these actions. There is a decreased growth rate. There is infertility in the animals because of abortions in the animals. Again, economic loss to the countries because of these actions. In case of animals, after recovery, there are very less chances of the recovery, but if it is there, reduced working capacity observed in case of a bullocks. There is a problem to the trade in the international market after such a types of epidemics in the country. As you know, after infection, we can summarize how various types of a clinical symptoms appear in the animal. There is a high grade fever, loss of appetite, dullness in animal. There is a profuse salivation. Vesicles are mainly found in mouth and mainly on the gums as well as on the tongue. This result ulcers and wounds on the hoof in the interdigital space, as well as the abortions in pregnant animal, and lastly mastitis like conditions. Here how this disease transmitted because of a feed, because of a contaminated water, as well as it is airborne type of a infection. So close contact with infected animal, because of a animal movement, the transmission will be very easy. We can prevent this infection by vaccination of all eligible cattle as well as a buffalo population. regularly twice a year is very important. It takes 10 to 15 days to develop immunity after vaccination. So, this is all about foot and mouth disease, the animals. Thank you. Thank you very much.